Yes, good evening. So, let me first of all know if you have any doubts for the previous session. If any doubts are there, you can ask me. Otherwise, we will continue with the next part. Okay. So, let's start with that. So, I'm sharing my screen. So, as I have told you, yesterday we were just having the introductory part, right? But from today, we will be having the actual practical sessions related to the databases, like how we can retrieve the data from the databases, and then we'll be using certain tools also for the analysis purpose. Okay, so try to attend these sessions properly so that you will be not missing any of the information. Okay, so as we have discussed yesterday, we are going to start with the database, which is what? NCBI. So may I know how many of you knows about the NCBI? Anybody have used this NCBI before? Okay. So basically, this NCBI is standing for the National Center for Biotechnology Information, which is having the information for a different things, like it is having the information for the gene, it is storing the information for the protein, for SR, like FASTQ format also it is storing, which is what? Raw data. So it is basically having a lot of information to be stored inside it. Okay, so we should know how we can retrieve this information. So that's why we are starting from the NCBI. That too, in the NCBI, we will focus on the GenBank database. But before that, we should know about the NCBI, right? So yes, that is what the first database we are going to deal with. And here what we are going to do, we will be having some theoretical part, how the data is actually stored in this NCBI. Then we will be getting familiar with the each and every option in the NCBI. And then we will be having some exercise questions where we are having one gene example, and we'll try to tell you how you can retrieve the information related to that one gene. Okay, again, retrieving the information is not that much difficult, but retrieving the correct information is difficult part, right? So let's go for the, without wasting any time, we will go for the next slide now. So basically in the NCBI, we are having one GenBank database. So this GenBank is a comprehensive public database. Can anybody please tell me what do you mean by public database?
Open access.
Yes, exactly. I think Ajisha ma'am was also answering.
Yeah, the same. Everyone can access the database.
Exactly. This NCBI is a primary database, means it is storing all kind of information. So here in this NCBI, it's not about the GenBank only. This NCBI is a public database, means if you want to retrieve the information, you can also retrieve it. I can also retrieve it. So it's a publicly available information. We don't have to pay for the money, right? So that's what public database. We can access the data freely. So yes, it's a public database of nucleotide sequences. Nucleotide sequence in the sense, what? Adenine, thymine, guanine, and cytosine, isn't it? DNA and RNA sequences. You can get it from the GenBank. And it is supporting the bibliographical and the biological annotations. Now, what do you mean by annotation? Annotation is nothing but any kind of extra information. Okay, so for that nucleotide sequence, if you are having any gene name, if you are having any gene ID, maybe you are having the gene function. So anything rather than the sequence is nothing but the annotation, right? So that extra information also you are going to get it in the GenBank database, and I'm going to tell you how that extra information look like. Okay, so basically this GenBank database is developed and maintained by the NCBI, which is National Center for Biotechnology Information. This is how the NCBI will actually look like. So basically, this GenBank database was created in the 1979 at the Los Alamos National Laboratory and was called the Los Alamos Sequence Database first. But later on, it was renamed as a GenBank in 1982 and it became the public database then. Previously, it was not a public, but yes, later on from the 1982, it became the public database, right? So yes, this is how the NCBI homepage will look like. We are going to discuss about each and every option over here. So before that, let's see what is this GenBank and how the information is stored inside that. Okay, so GenBank is one of the primary database. What is primary database now? Public database, we got to know that. Public means what? Anybody can access the data, right? But what do you mean by primary database now? Ayush sir, do you want to say something about the primary database? If you're not comfortable in unmuting yourself, you can just answer in the chat box. That is also fine for me. The first to refer to. Mm, okay, but it's not an exact meaning actually. It contains unprocessed data. Okay, okay. You're somewhere correct. You have given the 50% answer correct. Okay. Anybody else wants to answer? Okay, so basically, primary database means what? As it's a public database, anybody can submit the data, right? And as anybody can submit the data in this database means what? You will be having the experimental data also available in this primary database. Even if that experimental data is not correct, it's not cross-checked, still you can get that data in the primary database. Means what? You will be having the experimental data also in the primary database and you are going to have the cross-check data or the cross-verified data also in the primary. Okay, so basically this GenBank or the NCBI is storing the experimental data which is not cross-checked and the cross-check data also which is what? Validated data. Okay, so if you are talking about the secondary databases, so there you will be having only the cross-check data. It will be not having any false data, like any experimental data, you will be not able to find it out there. Okay, so this GenBank is one of the primary database like a EMBL and the DDBJ. These are also some another databases, primary databases, another primary data. Then the content in this GenBank is going to include the genomic DNA, mRNA, cDNA, raw sequence data also. Again, a lot of data is present, but we are going to see it in further while performing the exercise questions. Then in the GenBank, the text-based search method is used. Can you please tell me about what can be the sentences standing for? What can be the text-based search? Okay, so basically, whenever you're retrieving the information from any databases, we are having basically two types of searches. Like these are the main types of searches. Okay, you may have more also in another databases, but two basic types of searches we are having as a one as a text-based search And the second we are having as a sequence-based search. Okay, text-based search means what? If you are having any gene name or organism name, you are directly taking that keyword in terms of text and you will be retrieving the information from the databases. You are directly having the uh, search bar where you can provide your text-based keyword. Okay, then you are having a sequence based search. Means what? If you're not having any of the extra information, like you're not having the gene name, you're not having the gene ID with you, right? So how you can search for that information, right? If you're having only nucleotide or the DNA sequence with you. Okay, only sequence is there, but you are not aware about organism name or maybe gene ID or maybe related to the gene name. So there are some databases are there where you can directly provide your sequence and you can get the information. So as of now, our NCBI database is allowing only the text-based search. We can directly provide the search terms in terms of text. Sequence-based search is not allowed there. Okay, so I hope these types of searches are clear to you all. Shall I move to the next point now? Okay, so now yes, as we have discussed about it, then as we are going for the GenBank database, obviously each and every database will be storing the information in a different way, in a different file format. So from the GenBank, if you are downloading the data, so that file will be having the extension as a .gb and it will be having this much information. It will be having the header, it will be having the feature where the annotation information will be there. And in the last section, you will be having the actual sequence of it. So here we have provided one GenBank format, which you can see it here. So first of all, you are having the header section over here, which is storing the information about your maybe if you are searching for the gene, so gene information will be provided. So whatever the search term you are providing, for that you will be having the details along with the experimental information also. Then in the feature, you are going to have the different features. If you are searching for the gene, so in the gene, you will be having the exons, you will be having the introns, you may have the transcript, you may have the CDS region. So likewise for the gene, whichever the components are present, that will be provided here in the features. And then here at the end, in the third section, you will be having the actual sequence of that particular search. Okay, so yes, whenever we will open it in NCBI, you will be able to get it more clearly. Okay, so that is what just the introduction part we are having it. So let's get familiar with the NCBI homepage directly so that it will be more clear to you. So I'll just open this NCBI database. For that, what you can do, you can just go to the browser and just type it there as a NCBI database. Enter it, and then the first link itself will be for the NCBI. Okay, you just have to open this first link. I'm just pasting this link also in the chat box. Okay, so you can also open this NCBI into your system. Now let's try to discuss about this part. So basically, this NCBI is a, you can say it's standing for the National Center for Biotechnology Information, which is developed and maintained by the National Library of Medicine and the NIH. Okay, so now you can see like it's a primary database, that's correct, but it is a collection or it is a repository for a lot of databases. Like NCBI is having a lot of database to be interconnected with each other. Okay, so if you're searching for the gene information from the gene specific database, it will retrieve the information and then it will directly give it back to you. So how you can find which another databases are interconnected with this NCBI. So here in the all database section, just give me a minute. So now basically here, how you can find out which another databases are interconnected in this NCBI. So here you can spot all database drop down menu, right? So you can just click on that and here they will just enlist which databases you are interested in. Okay, so maybe if you want to retrieve the gene information, you are having a gene database, that directly you can select it and you can just be specific about your gene information. If you want to get the information related to maybe protein, so you're having a different protein databases also. If you want to get the medical subject headings, like this is also one database which is storing the medical information, so that also you can get it. If you want to go for a complete genome, then genome database is also mentioned. So whenever you want to retrieve the information from the NCBI, you have to be specific about this database section. Okay, so you can just choose them according to your like aim, or maybe according to your interest. Okay, then here you are having one search bar where you can provide your search terms or the keywords. Again, the keywords we have to provide in terms of text based search only, right? Maybe if you are having the gene names, or maybe if you are having the gene IDs or organism names, that you can directly provide it here and you can search for it and you can just get the data. Okay, then here if I will scroll bit down, here we are having this middle page. We will get back to this middle page again later on, but let's discuss about these panels now. So here if you will see in the left hand side, you are having some resource list, right? Resource means what? Again, from which sources we are getting this information, right? So again you can call this resources as an filters. Okay, so maybe if you want to be specific about your DNA or the RNA information, you can select that resource from here. Maybe if you want to go for a literature search, you are having a literature option. Literature in the sense, if you want to retrieve the research paper information, we are having a literature for that. If you want to go for a homology modeling, that is one CAD part. So yes, different resources are being mentioned. Again, you can choose them and you can be specific about your information further. So here you can click on the A to Z and again from like all the resources you can get it here. Then here, out of all this A to Z resources, people are using most of the time this particular resources. So that's why they have enlisted it separately. So you can see most of the time this PubMed database is used. So do you guys know what this PubMed is famous for? Anybody? What is PubMed? For biomedical literature? Yes, that's correct. Only one person is responding. What about the others?
To get the literature review.
Exactly. Yes, that's correct. Whenever you are searching for the research paper or the literature search, you can directly go for the PubMed. So yes, PubMed is also developed and maintained by the NCBI, like it is interconnected with the NCBI. So as people are using more this PubMed, so that is highlighted here in the popular resources separately. Then you can see the another options are also being mentioned. Then here you are having a NCBI news and blog section where the things are being mentioned, like if any news related to the NCBI will be there, if they are updating some another databases over here, or maybe they are giving you certain workshops or webinars, and they'll just update these things over here in the NCBI news and blog section. Okay, now let's get back to the basic part of here. That is what the middle part. So can you guys please unmute yourself and you can just tell me what comes to your mind whenever you are reading about the 6 boxes or the 6 points. What can be the submit, download, learn, research, analyze, develop options for? You can maybe unmute yourself, or otherwise in the chat box also you can just tell it to me in one line. Let's make it interactive.
Steps of data analysis.
Sorry, ma'am, your voice is actually low. Can you please adjust your mic?
Yeah, steps of data analysis.
No, actually.
No, okay.
For the analysis, we are having the analyze option, okay, specifically. Yes, sir. Any guesses from your side? Anand, sir. Like actually, it is not having any specific information, or it is not having any specific definition. From the word only, we can understand what it can be standing for.
With the summit, you are able to upload any data or any manuscript for publication, is it?
Yes, exactly correct. Download. Then download option we are having.
To download any data you are interested in.
Yes. Okay, I'll tell you the things. So basically, as we know that NCBI is a primary database, right? And it's a public database also. So anybody can submit the data, anybody can download the data, isn't it? So if I'm performing any experiment work and I want to submit my data also in the NCBI, so I can directly submit it from this section. Okay, maybe if I want to submit my manuscript also, so that also I can submit it. There will be some formalities that we need to fill, and then we will be able to publish our data. Download option is there. So as everybody is submitting the data, obviously we can download that data also in our system, right? So that you can download it from here. This option is also provided. Otherwise, directly we can just go to that page and we can download it. We are having the downloading options in a different way also. Then we are having the learn option. So basically, if you want to learn anything from the NCBI, so they are giving you certain documents, they are giving you certain tutorials or webinars or seminars. So if you want to learn anything from the NCBI, you can directly come to this learn option and you can attend their seminars, or maybe some video tutorials will be there. You can just attend them and you will be able to learn anything. Okay. Then we are having a develop option over here. So this develop means what? NCBI is already having its APIs. APIs are what? Application programming interfaces. So you can just like already they have developed some applications. So we can directly download their applications and we can uh, develop our own applications according to that NCBI. Maybe we can modify their applications and we can integrate it in our systems likewise. So that is what the develop. You can just develop your own applications or the libraries. Then we are having a analyze option. So obviously this NCBI is coming with a larger data, so we can analyze that data also. And again, if it is coming with the biological data, we know that biological data is two-way larger, right? We cannot analyze it manually, right? Maybe if you are having any sequence with you and you want to design the primer for that, are you going to do it manually? Can you design the primer manually? Okay, maybe you're new to the field, so maybe primer thing might be not known to you. So, okay, that is one biological term which is used for the amplification purpose. But whenever you are having the biological data, you cannot analyze it manually, right? So, for that, you have to use certain tools. And NCBI has already developed some tools which you can directly use for the analysis purpose. And among that, we are having one blast tool that also we are going to see. Okay, maybe if you want to go for the gene prediction, so for that also the NCBI has developed the primer 3 tool. A lot of tools are being developed by the NCBI which we can use for the data analysis purpose. Okay, then last option we are having as an research. Can anybody please guess now about the research? Ayush sir, Janvi ma'am? See, yesterday only I have told you the session should be interactive, right? So why another people are not responding? Only 2 to 3 people are answering the things. I'm not asking about the specific answers, but at least you should give me some kind of input from your side also, right? So to look for any new or the latest research or findings, the scientific community. Okay, okay. So as we know that we can publish our data also here, right, in the NCBI. So maybe if you want to collaborate with this NCBI, okay, like if you're performing an experiment and you want to take the help of this NCBI researchers, so you can just come to this research option and you can collaborate with the NCBI. Okay, you just have to submit your ideas for your experiment and your aim of the experiments. And if they will find it interesting, they'll directly give you the guidance for the same. Okay, so that is what the research option you're having it here. Okay, so yes, that is what the homepage of the NCBI looks like. This is what we are having it here. So if any doubts are there in this homepage, you can ask me now. Otherwise, we will directly go for the exercise questions then. Okay, we are having certain gene examples. We will directly take them and we will go for the exercise questions.
Excuse me, this NCBI and PubMed, like NCBI is only for the data, right? Data retrieval, and
No,
PubMed is for the literature review.
okay, ma'am. So
How are
basically,
both connected?
yeah. So basically, this NCBI is a collection of a lot of databases also. Okay. From the NCBI, you can download the nucleotide sequences information also. And as PubMed is also interconnected with the NCBI. So if you will, here in the all database section, if you will select the PubMed, you can retrieve the research papers also from here. Just the thing is
Okay,
that the information will be loaded in the NCBI directly from the PubMed database. They'll take it from there and they will update it here. Like,
okay, okay, fine.
and as you were talking about, I think, sequences from the PPT. I hope you have took that point from the PPT. So that was about the Gen Bank and not about the NCBI.
Okay, fine.
Yeah. So, okay, then if any another doubts are there, you can ask me. Otherwise, we will go for the exercise questions, okay? You can check it out this homepage. If any doubts are there in any of the option, you can ask me. Okay, let's go for the exercise questions then. So here we are having certain set of questions. Let's go for it. So try to perform it along with me. If any doubts are there, you can then ask it to me. Okay. So as I have told you in the NCBI or in this GenBank database, we are going to take one example of the gene and we will try to retrieve the possible information related to that gene, right? So here, first question we are having to search for a gene PRNPC. What keywords or what information you can get it from the question? As they are giving you the gene means we want to retrieve the gene information specifically, isn't it? So here in this all database section we can just select the gene database from here, isn't it? Because we want to be specific about the gene information. Then we are having the search term or the gene name directly as a PRNP, which is nothing but our text based search type, isn't it? And then we are having the organism name, which is Homo sapiens. Now let's provide this PRNP in the search box and let's search for it. Can you please search for it and let me know how many results you are getting it for the PRNP gene? Okay, okay. 16501145. Okay, I'll search for it. We are having 1650. Maybe some kind of filters you might have applied, PLE ma'am. Try to check it once. Okay. Now let's discuss about this particular part. So again, in the left hand side, you are having the filter panel. Maybe if you want to get the information for a non-coding sequence only or for the annotated genes only, that you can apply the filters and you can minimize your search. Then yes. Then in the right hand side, you are having the result by taxon. Means if you want to be specific about your organism, okay? So that organism names are being mentioned over here. You can see out of this 1650 results, we are having it here. 1145 are for the Homo sapiens. Okay, so Piali ma'am has given the answer only for that. Maybe um, only for the Homo sapiens directly. Okay, that's fine. So for the Homo sapiens, we are having these many entries. For the musculus, that is what mouse, we are having 79 entries. So from here, from this result by taxon, you can directly select your filters for the organism and you can be specific about your entry, right? So if you will see below, this is how the result table will looks like. So we are having 1650 entries. Among that, 20 entries are being provided in each of the page. This is what the first page. Likewise, we are having the 83 pages here. So first you are having the name or the gene ID. So you are having the gene name and then you are having the gene ID. Means if you are searching here in this databases, so for each and every gene you are having a unique gene ID that you can get it here. That is 5621. Okay. If you are searching for any protein information, again for each and every protein you are going to have a unique identifier. For each and every transcript, for each and every exon also you are having a specific identifiers. Okay, so here we are having the PRNP gene name and the gene ID for that PRNP gene. Then this is what the description we are having for the PRNP gene. This is what the location. So on the chromosome number 20, from this coordinate to this coordinate, your PRNP gene is present. Means here you are having the start and the end coordinates for your gene. Aliases are nothing but the synonym names to your PRNP gene. MIM is nothing but the another database. From there also they are mentioning the ID. If you want to get the details about it, you can just provide that ID and you can get the information in the MIM also. Okay, so here in this results, you are having the information related to the all organisms, but we want to be specific about the Homo sapiens. So either you can apply the Homo sapiens filter from here, and then you can choose your entry. Here also in the result page, first entry itself we are having for the Homo sapiens. So you can just choose this entry, you can open it and you can get the data. Otherwise, out of all these entries, people are searching more for the Homo sapiens only, so that's why here in the separate box, this entry is mentioned. Okay, this first one is the same. Okay, so you can open this entry from here also. That is also fine. So I will open this entry and let's try to discuss about this part now. Okay, so now this is what you are having a full report, which is nothing but the full information for your PRNP gene. Now here, if you will see in the right hand side, you are having a table of contents, which is nothing but which is containing actually the headers of the information which is already present in your main page. Now what happens, actually a lot of data you are like we are having it here. So if you are searching for the data manually, you will be not able to find out that accurately. You have to scroll a lot. So that's why the headers are being mentioned over here. Directly you can click on that and they'll just update that information likewise. So yes, you can call it as an index also. It's totally up to you. Then let's talk about the full report. So here, first of all, you are having a gene symbol, which is PRNP. Then you are having the description for your entry, where you are having a protein name, Kanu blood group, and Homo sapiens, that is for the organism name. This is what the description for your entry. Then again, you are having a specific gene ID for your PRNP gene. Then you are having the date at which the information was updated. So now here you can see we are having the summary for our gene. Okay. So first of all, we are having a official symbol, which is what PRNP, right? So this particular gene symbol is being provided by the HGNC. This HGNC is nothing but the Hugo Gene Nomenclature Committee, which provides the unique names to your gene. Hello. Okay. Then we are having the official full name. Here, this is just the symbol for your gene, and this is what you are having a full name for your particular gene. That is what prion protein Kanu blood group. And this is also provided by the HGNC, which is Hugo Gene Nomenclature Committee, which provides the unique names to your gene. Okay, primary source from which this information is coming, this is also from the HGNC. Then you are having a C related option. So can you guys please guess what can be the C related option can be standing for? This particular thing, what it can be?
Ensemble database, another database.
Yes, exactly correct. So basically, we know that NCBI is interconnected with a lot of databases. So if you want to get the TRNP gene information, maybe in the Ensemble database, in the MIM database, or the Alliance Genome database, they're directly giving you that links. Like you can just click on that IDs and you'll be like they'll directly update that information over here. Okay. Then we are having the gene type. This PRNP gene is going to code for a protein. So it's a protein coding. Then we are having a REF6 status. So REF6 is nothing but the reference sequence, which is again one database, which usually gives you the status of your entry. Okay. Now what can be the status? Can anybody please guess it? As I have told you about the primary database, you can link it with that.
It is cross-checked.
Yes, yes. Actually, we know that NCBI database is a primary database, right? It is storing the experimental information also, and it is storing the validated or the cross-check data also, right? So this RefSec status is going to give you that information. So if like I have performed some experiment and I have published my data, so RefSec status will be here experimental or the predicted. But here, whatever the information we have opened, this is what we are having as an reviewed. Okay, reviewed means what? This information is cross checked by the expert panel. We can completely rely on that information. Okay, then we are having like again, whenever you are retrieving the information, try to go for a reviewed entries. Okay, you can completely rely. Experimental data can be false also, right? Then we are having an organism from which this information is coming. Lineage is nothing but you are having a hierarchical information from which order, from which taxa actually it is coming. Likewise, then you are having the also known as, this is nothing but the synonym names to our gene. So our PRNP gene is also known as by these many gene names. Okay, then we are having the summary information where we are having details about this PRNP gene. Okay, so like from what is its function on the chromosome, what is its location, then if it is its expression is not proper, then which kind of diseases it is going to cause. So that kind of information will be updated here in the summary section. Then further we are having the expression option. So can anybody please tell me what can be the expression now? Expression.
Ma'am, the gene varied functions like the protein expresses for that gene.
Exactly correct. Yes. We know that as we are having a gene, so obviously in our body, somewhere it is going to express, right? So that expression we are going to get it here. Okay. So this PRNP gene is expressed into the brain, then ovary, and how you will be able to know how much it is expressed with the help of this RPKM count. So basically, this RPKM is nothing but the expression count. Okay, reads per kilobase million. So higher the RPKM count, higher is the expression. So yes, basically this PRNP gene is broadly expressed in the brain. Okay, 24 more tissues are also there in which this PRNP gene is expressed. If you want to get that details also, so you can just directly click on the see more option and you can see the graphical representation, like graph will come for that. So now here you can see RPKM count along with the samples. So obviously this count, expression count also they are going to identify with the help of certain techniques, isn't it? So there is one technique which is reference-based RNA-seq. It's an NGS application actually. So with the help of that NGS application only, we can identify gene expression in a different tissues. Now here you can see, if you will hover on this particular plot, you can see in the brain the RPKM is 144, and yes, the graph is also showing that in the brain only the PRNP gene is expressed more. Then if you will go for the least count, so in the pancreas you can see we are having the least expression of this PRNP gene. Okay, and then they are giving you the experimental information also while counting this expression of this gene inside that tissues, how many number of samples they have to count, what quantity they have got likewise. Okay, so this is what we are having the expression option. Okay,
Ma'am, can you
yes.
repeat the full form of RPKM?
Okay, it's a reads per kilobase million.
Okay, ma'am.
Yeah. Then we are having the orthologs section. Now, can you please tell me about the orthologs, what it can be?
Same gene, different species, something like that.
Yes, exactly correct. So orthologs is nothing but the same gene in a different organisms, but the parents are common. Likewise. Okay, so that organism names you can get it over here. Like the PRNP gene is present in like same gene, different organisms, but parents can be similar. Likewise. Okay, so that you can click on the all and you will be able to get it. Then in the gene table, you are going to get the information related to your gene in count format. Okay, as it's just the table, you will be not having a detailed information, but you will be having certain information in terms of IDs. Then here you are
Excuse
having,
me.
yes,
This parents similar means
sorry.
similar parents means that the
Okay,
parents should be of the
okay.
same organism.
Either your gene can be expressed in, maybe it can be expressed in a alpha chain or maybe it can be expressed in a beta chain. Okay, so if it is alpha and beta, these will be nothing but acting as a parents. Let me just show you one image so it will be clear to you.
Okay.
Mm, see, so here they are giving the examples related to the globin gene. So here you can see globin gene can be expressed in alpha chain or beta chain. Okay, so this will be serving as an parent over here. So if your gene is getting expressed in alpha chain, it is expressing in a frog alpha, human alpha, mouse alpha. Try to remember about these chains. If your globin gene is expressing in a beta, it will be expressed in a mouse beta, human beta chain, frog beta chain. Okay, so now here you can see same gene, but if the organisms are coming from the same parent, okay, same gene, different organisms, same parent, that is what the orthologs. If you are having the same gene, different parents, different organisms, these are paralogs. And together this orthologs and paralogs will comprise the homologs. It's irrespective of the parents, only the gene, but different organisms likewise. Is that clear now?
Yes, yes, ma'am.
Yeah. So then we are having a gene table and the transcript table. I'll just open this links in the new tab so that you'll be able to know. Maybe you're not aware about the transcript concept. So yes, I'm giving you the like assignment. Maybe you have to read about certain topics. So try to finalize that because that will be required tomorrow. I'm not going to go it in detail. I'll just give you an overview about it. So try to read about the what is the actual structure of the gene, how it like how the gene is made actually. Then read about the transcript, read about the CDS region, okay, and that's it. So read about these topics, okay, so that this transcript table I will tell you tomorrow. Okay, then uh, the next question we are having, on which chromosome it is located. So again, if you will see it here, in the genomic, like here in the summary also it is mentioned as it is present on chromosome number 20. In the genomic context also you can see the chromosome 20 is mentioned, and here your exact location of the gene is mentioned, isn't it? So exact location in the sense, from here we can identify our gene is present on the chromosome 20, but on the chromosome number 20, at which point, at which specific point our gene is located. So that we can identify from this location. But it is present in some coded format, right? It is 20, we can understand it's a chromosome number, but what is this P is standing for? What is this 1? What is this 3? Anybody knows about it? Any guesses?
Ma'am, chromosome has two arms, P arm and Q arm. So
Yes,
it is located on the P arm at position 13.
at position 13. Okay, do you know the actual meaning for the 1, 3, that 13? It is
No.
also having certain information actually. Okay, I'll tell you the thing with the help of one image now. Yeah, with the help of this image, we can understand that. I hope this image is properly visible to you all. So here you can see they have took the example of the CFTR gene. So as correctly said by the sir, in the chromosome, we are having a short arm and we are having a long arm, right? So here you can see we are having this short arm and here you are having the long arm. So according to that, at which actual arm it is present, so that you can identify. So the code is like our gene only. So 7 is nothing but the chromosome number. Q is nothing but the long arm. If your gene is present on the short arm, they will represent the code as a P. Okay. Then here they are giving the Q means this particular CFTR gene is present on the long arm. Again, in the long arm, we are having certain regions, certain bands, certain subbands. So this 3 is representing the region at which it is present. 1 is representing the band. And whatever the number after this digit is there, that is nothing but the subband.
So, ma'am, 1-3 implies only the region and the band, not the subband, right?
Yes, exactly. So here if you will see our gene location, it's a 28th chromosome. P is what? P is nothing but your short arm. And then 1 is nothing but the region, 3 is nothing but the band. And as dot and further thing is not mentioned, so subband is not given. But yes, till the band, the things are provided. Okay, so I hope this location part is clear to you all. Okay, so I think that's enough for the today's session. Try to go through from this NCBI database, and tomorrow we will be solving this exercise questions. I have given you certain points also to read about. So read about that points also. Okay, so I'm stopping my screen share and I'm just sharing the attendance link with you all. Try to fill it. Okay, I have shared the attendance link. Try to fill that. And let me know whenever your attendance will be done so that we can end up the meeting. And let's meet tomorrow for the next exercise questions.